probability of any given match was P. That's the probability of matching at a single. Actually, I think I was using M when we talked about this. The probability of any single match is M. So what's the probability that these two random sequences match completely? Two sequences, length M. Every position is picked randomly from an alphabet of size Z. P equals 1 over Z. So the probability of a match at any given place is P. All right, what's the probability that they match in their entirety? Yeah. No, just scratching. Okay. What's the answer? P to the M. Okay. So P to the M is the probability of a complete match. And as a function of M, that falls very rapidly. Okay? So I don't know really how to draw this, but as a function of M, P to the M falls very, very rapidly. Okay? But what did we get when we asked a different kind of question? When we asked if we have a huge sequence here, then, and we still have the same M there, we asked, we were interested in what's the probability that it matches completely somewhere. Okay? So the probability of a complete match, call this thing Q, call this D, of Q in D somewhere, or at least once. Okay? So how did we analyze that? We said at any given position, we know what the probability of a complete match is. That's P to the M. So the probability that it didn't match there is 1 minus P to the M. Okay, that's the probability that at a given position we didn't have a complete match there. Therefore, the probability that you never have a complete match is that roughly to the n. This is too big. It really should be n minus m, but n is considered to be huge. Okay, and therefore the probability that it does match somewhere is that. Okay. Now, what did we simplify that to be? Anybody remember? Well, this one we said is the same as n over n. Okay. And therefore, what does this converge to? It's about e to the minus npm. Okay, that's what this does. And then this is 1 minus e. Okay? All right. And the point of this was that if you now make n big enough compared to m, and we actually studied what that trade-off point was, okay? When n is big enough, compared to m, then the probability of a complete match of Q in D, it's the database, the huge long string, is very high. Okay, now I'm being very vague here, but where was this big enough point? Anybody remember? Is what? Okay, 0.63 is where we used that in order to study to get our hands actually on a particular trade-off point. But we actually came out with a function finally on the relationship between that uses n, p, and m to say where we thought it was the probability was starting to get big, and the starting to get big was the 0.63. But so what did we do to for this? Okay, nobody remembers. Yeah, say that again, a little bit louder.
So m is about size of log
M is, yeah, about log of z or p? Z of n. Okay, and this is actually a little bit backwards way of saying this, but so in which direction does the probability get bigger and bigger? Where m is smaller than this or m is bigger than this? Yeah, okay. So when n is really smaller than log zn, then the probability, this probability, gets large. Okay? And actually, we had this we had it stated in a reverse way. We had n in terms of m. Okay? So I think it was when n gets to be bigger than basically the reciprocal of p to the m. Is that right? Then the probability gets high. My point, what's my point in all of this? Actually, there are 2 points. One is that if you just think about the probability of a complete match, one string of length m against another string of length m, all you can study there is what happens as a function of p, but p is fixed, and what happens as a function of m. And what you learn from that is that the probability drops off very rapidly. So you take the take-home lesson that if these strings are very large, m is large, the probability of a complete match is low. But over here we're saying, yes, okay, m is going to be big, but if n is even bigger, and we're being precise here about what even bigger means, then the take-home lesson is no longer that the probability drops very, very rapidly as a function of m. It really has to do with the interplay between n and m. And when we worked out some examples here, n doesn't have to be that huge, at least this is an intuitive statement. N doesn't have to be that huge in comparison to m before this looks like a reasonably good probability of a match somewhere. So these are very different kinds of things, phenomenon. What's the probability of a particular event, this complete match between this, and the probability of an event like that happening if you have many, many, many trials. Okay? And 10 years from now, if you forget everything else about the statistics and the probabilities that we did in this course, you should remember that those are intuitively very, very different. And I'm going to get onto a story in a minute that illustrates the danger of forgetting that. Okay, the second point I wanted to make was to remind you of this here. This is the probability of a complete match of Q and D somewhere is this. All right? So when it comes actually to being a little more precise, we can state something. I want to just put up, so you see this once in your life. The max of many independent, let me see where I'm reading this. Yeah, the max of independent random variables follows what's called an extreme value distribution. I'm not going to derive that, but the extreme value distribution has two parameters which are used to describe the particulars of it. And those are U and lambda. And the form of this is the following. The density function of the extreme value distribution basically actually that's not what I want. This is the distribution function. The probability of a score greater than or equal to x is the following. 1 minus e to the minus e to the lambda u minus x. Pretty awful looking thing. Okay? But kind of reminiscent of this one. At least, very superficially. And okay. And the density function, at least when I think lambda and u are 1, that density, let me see if I can find it. I put it up for you last time very approximately. Let me see if I can draw that better. Looks kind of like that. Okay? So this is again, the point I'm making. Has anybody ever seen this one before? Okay, good. The point I'm making is that this thing has a very long right tail. In fact, I didn't fully emphasize that even this part is pretty thick to the right of the mode. Okay? So it's not that unusual that you find a max, a large max, compared to the mean, or the mode in this case, of the distribution. But if you're thinking in terms of a single match, then as m gets large, that seems like a low probability event. And if you thought intuitively that this should be somehow normally distributed, where the normal distribution comes down very quickly, very close to the mean, then you're going to be drawing wrong conclusions. You're going to see matches that look, if you evaluated them according to this distribution, they look quite rare and unusual that they would occur by chance. But if this is the proper distribution, then it's not necessarily so odd. Okay, let me try to make this a little bit more, bring it back to biology. So one of the things I put on the web is some notes about BRCA1. Okay, so BRCA1 is a gene that, I guess this is almost 10 years old at this point, that people found mutations in that give a predisposition to breast cancer and ovarian cancers in certain subpopulations. And this gene was cloned, it was located, and the mutations in it were studied. So a great deal about it was known in terms of its genetics and relationship to breast cancer, but not very much was understood about how it actually worked and what it did. So what the geneticist that did was what we all do, what everybody does these days, is you take the BRCA gene. Actually, the biggest mystery to me about BRCA is why it's pronounced BRCA. When obviously, it's BRCA. I guess it's very hard to say. Well, anyway, however you would pronounce it, BRC with A, people pronounce it as if the A was there, or there was an extra A there. Anyway, so you take this sequence, and then you do a blast search of the sequence against all the existing sequences. And you also look in repositories such as ProCite. So in this class so far, we haven't explicitly studied ProCite, but I know I've mentioned it in one of the Pearl notes. So ProCite is a database, basically, of regular expressions. So we all know what regular expressions are. Where do these regular expressions come from? What do they mean? People have found a nice, for different families of proteins, where by family I mean a set of proteins that are related by some function or structure. They're related biologically. People for each family have found a regular expression that characterizes that family, that matches to the strings that are in that family, to the sequences that are in that family, and doesn't, at least not too badly, have false positives. It doesn't match to sequences that are outside that family. So ProCite, I can't remember whether I think there's an S in here, is a collection of these regular expressions, that each characterize some family of proteins. Okay? And so when you have a sequence and you don't know much about it, well, you run BLAST, you see what it matches, and you see what's known about the sequences that it matches. But you also use these kinds of specialized or more biologically informed databases as well. eMotif and a whole variety of other facilities that are available. So what happened in the story about 10 years ago was that, again, nobody really understood the functioning of this BRCA sequence. So they ran against ProCite. ProCite at that time had about 1,000 regular expressions in it. I think now it probably has maybe double that. So, and some of these regular expressions, if you look in the ProCite, and we have a lab where I'll ask you to do that, some of the regular expressions are actually quite small, and a lot of them have wildcards in them. So it's not that difficult that you're going to find the regular expression matching somewhere in a sequence. BRCA sequence is quite long. It's over 1,000 amino acids. Okay? Once you translate from the DNA. So what happened was you have this BRCA1 sequence, and here was the regular expression for a family of proteins. called granins. Okay? Now, let me just, for the sake of complete simplicity in this discussion, let's just imagine that this regular expression was just a string. It's not, actually it is a regular expression. It has wildcards like periods. It has some repetitions and so on. But just imagine, I don't know, I'm just going to make up some, something like that. And remember, what does it mean for this regular expression to characterize a family of proteins? It means that up until that date, if you looked at all the granins that were known, every one of them has this particular regular expression. It matches to that regular expression somewhere. So in my simplistic example here, every one of the known granins would have this motif in it. But if you looked at the other known protein sequences, very few of them that were not granins only a teeny number of them would have that. So this became a good characterization, a good characterizer of whether a sequence was a granin or not. Okay. Generally, ACQ would not have that. All right, so what happened? So they ran the BRCA sequence against ProCyte, looking for whether there was any regular expression that matched in the BRCA sequence. And they found this match to granin. Has anybody ever heard of a granin protein? Anybody study proteins here? It's very obscure, apparently. I'm not a good judge, but so the fact that I never heard of it previously means nothing. But I've asked this for about 10 years, and nobody really that I've met has ever said, yeah, I know a lot about them. So it's an obscure family, and it suggested, and you can read it in the notes, something very interesting possibly about the functioning of BRCA1 and its relationship to cancer. Okay? Then people started doing some experiments, and sort of the usual thing that happens early on in any kind of field. Some experiments said, yes, this does seem to be related to cancer. Others said, no, that doesn't seem to be related at all. And after 10 years, the not at all seems to have definitely won out. But where do the statistical issues come in? The people who published this paper that said that BRCA1 is related to granins, and it got a lot of press, big articles in the New York Times and popular magazines and so on, because of the sort of interesting way that this would suggest that cancer worked. They did that on the basis just of this search. They didn't really have any other intuition except that they got this nice hit. And they published a probability. They said the probability, well, this was the claim, the probability that BRCA1 would contain a granin, or contain a subexpression or substring matching the granin regular expression was, I think it was 0.00175. Okay? So that's a pretty small number. And we actually discussed a little earlier what kinds of cutoffs people typically use when assessing whether something is possibly occurring at random or not. And oftentimes people will use, 0.05 or 0.01. So this is a very small number. And based on this number, they were able to make an argument that this was a non-random event. The fact that the BRCA sequence had in it something that matched the Granin regular expression was something that would happen with a very low probability. Okay? And this number is right, roughly right. Okay. But the conclusion, well, some people at NCBI looked at that paper and said that the Granin motif has no statistical support. They weren't biologists or biochemists. They weren't saying that Granin had no role here, but they were saying that the statistics that these people did was all wrong. Okay? That this number didn't make any sense. You could not get from this number any statistical support for this being a non-random event. And they said that the proper number should be, the proper number to use, the proper probability that this is non-random. Well, sorry. The proper probability that this is random. I'm really abusing the language here. Anyway, the probability of a random hit of that quality is like 0.889, which is a big number. Okay, so there's a big difference between these two numbers. Can anybody see intuitively, back to my discussion about the difference between single experiments and multiple experiments, the difference between the extreme value distribution and the probability of matching a single event? Anybody see how that comes up in this story? And why there's this big difference between these two numbers? I mean to say it's not numerically wrong. It's not that they made a big mistake and they pushed the wrong button on a calculator or something. It's the correct number for a particular question. It's just not the right question.
Is it because for a single match we have normal probability distribution, while for an extreme match we have a more strict distribution, and it's harder to get more matches? Calculated it as a multiple event, but it was a single event, and it had much more probability to do that.
Yeah, I only heard half of what you said, but it had some of the right keywords. It had some of the right. This story would be a lot simpler in terms of explaining it, connecting it to what I've said so far, if the regular expression wasn't smaller than the sequence. So at least. Okay, so let's simplify the problem a little bit. Imagine that the regular expression was about the same length as the sequence. Okay? And imagine that what they'd computed was, what's the probability of a random sequence matching this? Okay, so you have a Granin sequence, however long it is. And you have a model that says your characters in your random sequences are picked uniformly, with some probability, 1 over 20, whatever. Or they probably use the correct distribution of amino acid frequencies. Okay? So if I have a particular amino acid sequence and I ask, what's the probability that a random string that I pick matches that? That's like what we computed earlier, this p to the m. Okay? And this can be very low. This can look very low when m is reasonably long, even just 10, 15, 20 long. Okay? Now, actually, this is a little more complicated because what what this was computing was what's the probability that a motif or a regular expression, the Granin regular expression, would match in a random string that was as long as BRCA1, okay, which did give you multiple possibilities. Okay? But it still was low. It was still quite low. But the true multiple experiment that they did, where this multiplicity comes in of starting positions or multiplicity of possible matches, is they didn't just match to Granin. They didn't have any intuition ahead of time that BRCA1 was related to Granin, and then did that comparison, found a match, and then asked the question, well, what's the probability that that would happen at random? Because then that might have been a relevant question. Okay, but rather, they did a match of the BRCA1 sequence to everything in ProCyte. And the relevant and without any additional biological intuition, the right question to ask was, what's the probability that a sequence, random sequence that looked like BRCA1, the same composition, same length, was going to find a good match to at least one thing in ProCyte? And when you do that, then you came out with this number. So the difference between this number and this number is this number asks, what's the probability of a random sequence that looks like BRCA1, same composition, same length, frequency composition, matching this particular string or matching this particular regular expression? And that's low. Versus what's the probability of a random sequence with this composition matching something in ProCyte that looked, let's say, as complex as this one? And if that probability is high, which it was claimed to be, then you have no right to feel confident that the match you found has some biological meaning. Okay? But that's the point I want people to understand 10 years from now, because that's a classic mistake that people make over and over again. Let me try to illustrate it with, if I flip a coin 100 times, it's going to come out with some sequence of heads and tails. And if I then ask, what's the probability that I got exactly that sequence? Okay? Well, let's say it's a half to 100th. It's a very low number. All right? But what's the probability I'm going to come out with a sequence with that probability? It's not only very large, it's 1. Okay? I mean, so imagine, I flip a coin 100 times, and I get some sequence out. I compute the probability of that particular sequence, and it's a very low number, 1/2 to the 100th. And then I say, there must have been some force. That's such a low probability that it must have been because of UFOs or, the tide or whatever that would have created. I mean, what's the probability of getting that particular one is so low that, must have been some non-random force. But if you look at the experiment in a little bit larger context and you say, what's the probability that I'd come out with that conclusion that some non-random thing was happening, you have to say, what's the probability that whatever I flip is going to have that low a probability? And that's 1. Any given sequence has a probability. Any given sequence, any particular given sequence, has the same probability, 1/2 to 100th, right? So since you're doing this experiment, this larger experiment, which has an absolute certainty, probability of 1, that you're going to get excited if you look at the probability of just what you got, then you have no right to be excited. Okay? And that's exactly what's going on here. And technically has to do with the difference between looking at the probability of what actually happened versus looking at what could have happened and looking at how likely it was that you're going to come out with something that looks unlikely. It's very likely that you're going to come out with something that looks unlikely. No matter how unlikely that particular thing looks, it's irrelevant. Okay? And this is a really key distinction that you have to keep in mind. And of course it comes up in, well, here we're giving it in the context of database searching, but it of course comes up in the context of blast statistics. Blast statistics do properly adjust for the fact that you're searching in a very large database. Therefore, you're doing many, many experiments, as it were, asking whether a particular query matches some particular string in the database or the next string or the next string or the next string. As your database gets big, the probability that it matches, even if it was random, gets big too. Can get big also. Okay? So I hope everybody at least gets this and avoids the avoids this trap. The person who did this was a famous geneticist. I don't know that they've ever recanted, but over the years it's clear that BRCA1 did not in any way behave the way that they were claiming. In fact, the people who did this statistics, Stephen Altschul and associates, are the same people who do the statistics in BLAST and came up with the proper formulas. And they went on, in fact, really, again, using the database in a proper way. They went on to figure out some of the biochemistry of BRCA1, even though they don't know any biochemistry. They just knew how to use the database a little bit more appropriately. And one of the things they did, for example, and this is one of the things that everybody should do, if you use something like ProCite, and it says, ProCite comes out and says, why don't you think about BRCA1 maybe as a granin? Okay. Then you should think of this as a filter, not an oracle. Those are crucial distinctions. A filter sort of tells you something might be interesting and throws away some other things that might not be interesting. An oracle tells you the right answer. Okay, you just believe it and go on. So once a filter has told you this might be interesting, take a look. Then one of the things you want to do is a full alignment. Don't just rely on the regular expression that characterizes the granins. Start taking a bunch of granins as sequences and see whether they align well to the BRCA1 sequence. You should have more. You should have a reasonable alignment there. Maybe one that wouldn't have shown up in BLAST or wouldn't have shown up very powerfully if you just do BRCA1 against a single granin. But you ought to start seeing good alignments, good local and even some good global alignments to this sequence between granin sequences and this sequence. The other thing you want to do is, I forgot, but it's in the notes. There's one other thing that they did that made sense. It had to do with where these matches were occurring. Okay, but anyway, this is the key question. And if you look on the midterms and the final exam, the kind of questions that I ask, at least in this class, I don't just ask purely technical questions, calculate this number, give me this formula, and so on. I ask questions like, what's the take-home lesson of this story? What is it that you learned from this that you're going to remember? Why is this wrong? Why, in your own words, why is there this discrepancy? Okay, and so on. So I hope you can, internalize what that was about. So on the web, I have the notes about BRCA1, but I also have notes about database searching, which again I think make some of the same points about the differences in multiplicity. This difference between multiplicity, or the difference, okay, there are two issues involved. One is the difference between doing a single experiment and asking what's the probability of that outcome versus doing many experiments and asking what's the probability of at least one outcome that looks weird. That's one theme. And the other theme that's going on in here is, maybe it's the same thing, If you do an experiment and then you get a result out, and you ask what's the probability of that specific result, that's usually the wrong question. That's the question that most people ask, and it's usually the wrong question. And it comes up all the time when people make wild inferences. You know, you're walking across campus, and you think about somebody, and then you see them. Okay, actually, it happened to me just today. I was on the receiving end. I saw somebody stop to talk, and she said, "I was just thinking about you." What's the probability of that occurrence happening? I don't even know how you cast the right model, but if you start thinking about all the people you could have bumped into, and in what percentage of time do they think about you, and on and on and on like that, you're going to calculate it to be a very, very small probability. Okay, but what's the probability that that will happen to you once in the next 10 weeks, okay? Or something, an event that seems equally unlikely, like that, is actually, if you try to put the model properly, a very high probability. So you have to draw different conclusions. If you're thinking of it as a low probability event, then you're saying there's some force that drew us together, or I'm very special, or I don't know what. And if it's the other model, this is just some random event, then you draw a very different kind of conclusion. And that's the larger moral here that comes up very, very clearly in database searching and deciding whether you found something weird when you found something. I think I talked about the birthday paradox, didn't I, in this class? I vaguely remember doing it in this room, but it may have been in a different class. Okay, so again, let me give you, this is a really concrete example. How many people have seen the birthday paradox in a probability class? At least some of you, okay? So here's the thing. We ask each person what's their birth date, not the year, okay? And we're interested in whether or not there are two people in the room who have the same birth date. So the first question is, well, obviously, as the number of people increases, the probability increases. In fact, if you get above 365 or 6.
I remember I heard of them in the statistical thinking course in I think statistics 10. I think Dr. Utz in statistics 10 talk about the exact number. I don't remember probability.
Right.
But that's the resources that.
Yeah, we can figure it out right now. But the point is, okay, so as the number of people increases, in fact, as it goes higher than 366 or whatever, the probability of two people having the same birth date is 1. Okay? So the intuitive question is, how many people does it take before the probability gets to be half? Well, a lot of people would say, let's see, 365 divided by 2, what is it, 180, 182, 183, something like that. Okay? In this room, we have probably 45 people. Okay, way lower than half. I would be willing to bet something. If we start here, because it's never failed, whenever I've done this, you start here, we'll probably get a match, but before we get half the room. Okay, and the right number is that the probability gets to be above a half, I think, at around 33 people. Okay, so if I pick a particular date, July 10th, the probability that somebody here has that birth date. Does anybody have that birth date? It's about 1 over 365. It's a low probability. But if I ask what's the probability that at least two people have the same birth date, and I haven't told you what particular day we're talking about, when you get above 33 people, the probability gets above a half. And it's, again, the same phenomenon over and over again. And if you've never seen that in action, you should try it next time you're in a group of people with about 30 or 40 of them. All right. There's a midterm on Monday, and see you then.